So we're going to start this video with transpiration rate graphs. We're going to have a couple of different factors that will affect our rate of transpiration. We're going to start with light, wind, temperature, and humidity. So let's actually start down here with humidity instead of in the order that I read them. So with humidity, we have our rate of transpiration in the y-axis and our relative humidity in the x-axis. So as our relative humidity increases, our rate of transpiration is going to decrease. This is a result and this is a result of the This is a result of the the steepness of the concentration gradient between the water vapor, between the concentration of water vapor inside the leaf airspace and outside the leaf airspace. So we have the concentration of water vapor inside the leaf airspace is high, and the concentration of water vapor outside the leaf airspace in the atmosphere is variable. So the relative humidity outside the leaf airspace, if that relative humidity is high, that means that we will have a less steep concentration gradient, which means our rate of transpiration will decrease, which means we'll have a decreased rate of diffusion. So that means we'll have a low rate of transpiration, low rate of diffusion. A low relative humidity means that we'll have a dry atmosphere and a high level of a high concentration of water vapor inside the leaf, because the rate of evaporation of water off of the film inside the mesophyll of the mesophyll in the leaf is going to be the same. So we're going to have a high concentration gradient, a steep concentration gradient, and that will be an increased rate of diffusion. So an increased rate of diffusion yields an increased rate of transpiration. So that will give us a brachiostomic graph. Now, in our temperature curve right over here, we have our rate of transpiration in the y-axis and our temperature in the x-axis. As our temperature increases, our rate of transpiration is going to increase until we hit a stopping point. Now this looks familiar. This is going to look like an enzyme kinetics graph. This enzyme kinetics graph, it's not as a result of enzymes though. This is actually as a result of, first, the rate of diffusion as a result of the movement of particles, because high temperature yields high kinetic energy of particles, and high kinetic energy of particles is going to increase our rate of diffusion. So high increased rate of diffusion is going to increase our rate of transpiration. And that means that high temperature, high rate of diffusion, low temperature, low rate of diffusion. Then that means rate of transpiration is going to increase as well. We reach this plateau here as our stomata begin to close at high temperatures. High temperatures will close stomata. Stomata will close at these higher temperatures as a protective mechanism for the plant. The plant is going to close these stomata in order to prevent extreme water loss from that plant. Now, wind is going to be the same thing. It's going to look very much similar to the temperature graph. And this movement of air is going to be on our x-axis instead of temperature. This is going to be the same sort of idea as humidity though. Instead of kinetic energy fueling our rate of diffusion, it's going to be the steepness of our concentration gradient like humidity was. So air movement is going to take the water vapor in the atmosphere away from the underside of the leaf. So as water vapor diffuses from the inside of the leaf to the outside of the leaf, that air movement on the outside of the leaf is going to take that water vapor away from the leaf, creating a low water vapor potential outside the leaf. So high water vapor potential inside the leaf, low water vapor potential outside the leaf, and it's being prevented from reaching equilibrium by a mechanically removed water vapor potential. So we are maintaining an artificially low water vapor potential through wind. And this results in an increased rate of transpiration through the steepness of our concentration gradient being maintained. Now, as wind increases, the rate of transpiration will increase up to a certain point, because high wind, much like temperature, will also result in the closure of stomata. Now, light, we see a very different graph. We'll see light intensity as one of our curves, and transpiration rate as another of our curves, and time is going to be on our x-axis. So this is going to be an increased value. So this is just going to be a general arbitrary units of increased value. So as 0, 100 hours to 2400 hours to noon, 1200 hours, our light intensity is greatest at 1200 hours. Our rate of transpiration is highest slightly after noon. This is a result of a time lag. A time lag between greatest light intensity and greatest rate of transpiration. This is a combination of factors, both the greatest temperature of the day, as well as time lag between the light intensity causing enzymes to be made by the leaf. So light induces enzyme production inside the leaf. And those enzymes produced in the leaf are going to allow the stomata to open. That will allow for the transpiration to occur. So as the light intensity increases, stomata will open after a time lag because proteins are being produced, induced by this light intensity. As the heat of the day increases and the temperature rises, those proteins are going to denature, and also that temperature rising will also cause the stomata to close. So we will also see that temperature drop off during the day. Light intensity will also decrease, so we'll also see that our rate of production of those proteins is going to fall off as well. So we'll see a day-night cycle in terms of rate of transpiration, in terms of light intensity. So this is how we see this following of day-night cycle with light intensity and the time lag for transpiration. Now, for our potometers. Potometers are a piece of equipment that we can use to estimate water loss. And we do this by measuring the by measuring the decrease in the column of water over time. Now, a simple potometer water setup would be this U-bend PVC piping or plastic piping with a plant inserted in here. Now, all of this has to be done underwater. You cannot break the seal. If any bubble of water is ever introduced into the xylem, then a continuous xylem will be lost, and you will not have continuous flow of water through the xylem vessels, and you will lose the water transfer, and you will lose a transpiration flow. You will lose this transpiration pull. So effectively, you have an airtight seal and a water-filled tube and a calibrated pipette. Remember, calibration from Unit 2 is you have a series of tick marks that are measured against a known value. They give you a known value, that you have these measurements mean something. They have a known value associated with those tick marks. So at 0 time frame, the meniscus here has this tick mark. After 30 minutes, the meniscus is here, and that means that it has dropped by this many tick marks, which means this minus this yields us that many tick marks. I believe that's 2, 3, it's about 6, or yeah, it's about 6 tick marks. I'm sure that those watching the video at home have paused the video to count for themselves, but I'm not taking that extra step. So let's say that that's millimeters, and that's cubic centimeters, for example. So 6 cubic centimeters in 30 minutes. So that gives us a rate of 6 cubic centimeters every 30 minutes. That would mean that it is 6 cubic centimeters divided by 30 minutes. That is a rate in volume over time. So you could use that to get 6 divided by 30, 1 cubic centimeter for every 5 minutes. All right, that is your rate of transpiration. All right, or 0.2 cubic centimeters per minute. So that's your rate of transpiration, estimated water loss over time for this plant. Now, that's not really enough. We can do more about this. We can use a number of different potometer setups to measure a control. We can increase the misting inside by putting the plant in a bag and spraying the inside of the bag with water droplets to increase the local humidity around the plant. Or we can put a circular fan in front of the plant to simulate wind. Or we can put a bright light at different distances from the plant to simulate different light intensities. Or we can put the plant in front of different light filters to simulate different light intensities or different light shades, or different light or different color lights like blue, green, red, or different shades of light, etc. And then we measure the water loss in those different circumstances. And measuring those water losses in different circumstances will give you those sorts of factors. Now, what we can also do is we can measure the water loss per unit surface area of the leaf. By calculating the surface area of the leaf, what we could do is we could take a cutting of the leaf. We could take one leaf and we can put it against a graph paper grid, trace that leaf, and then count the boxes that that leaf takes up. And that will give us a rough estimate in cubic millimeters, or square millimeters, what surface area that leaf is. And then we take one of those square millimeters of that leaf, and then we put that under a microscope, and then we could count the number of stomata on the undersurface of that leaf, and then we could count the number of stomata per square millimeter. And then we can use that to determine rate of transpiration per stomata. Or we could do rate of transpiration per square millimeter surface area of all the leaves of the plant, estimated. So we can use this measurement to get quantifiable data. We can use these setups of transpiration to get quantifiable measurements. Now, these are the sorts of questions you should be able to answer. How is the column of water going to be able to respond in each of the photometers if the intensity of the variable being tested is going to increase or decrease? So if I increase the intensity of the circular fan, or if I move the light bulb closer or farther away, how will changing the surface area of the leaf, removing leaves, or covering leaves, or painting over the surface of a leaf with lacquer, how will that affect the rate of transpiration? Now, photometers will measure the rates of water uptake only. A cut stem is not the same as the uptake by root hairs. So the problem is that we can't actually measure and use this to put forward that this is the same as a plant in nature, because one of the problems is we have cut off the stem from the roots. We can't measure root pressure. We also can't account for different soil densities or the different concentrations of solute in the soil. So we can't account for those variables. Now, let's talk for a moment about the environmental factors, that affect transcription. Light is going to speed up transpiration by warming the leaf, opening stomata. It's also going to induce It's also going to induce transcription and translation. That's not listed here, but that is one thing it's going to do as well. Humidity. Decrease in the humidity will increase the transpiration because there's a greater difference in the concentration of water inside the leaf versus outside the leaf. Increasing wind speed is going to increase the rate of transpiration because you have an increase, because you're carrying the humid air near the stomata, carrying that away from the leaf, thus increasing the difference, the steepness of the concentration gradient. The soil water. If there is higher intake of water at the roots, or if the intake of water at the roots is not keeping up with transpiration, turgor loss is going to occur and stomata are going to close, and that will decrease the rate of transpiration. High levels of carbon dioxide in the air is going to usually cause guard cells at the stomata to close, and that will decrease the rate of transpiration. So high levels of carbon dioxide in the atmosphere will actually decrease rates of transpiration. This is possibly a protective factor against forest fires, which would very easily dry out the plant. Now, season. Although these videos are going to be filmed in Florida, so we don't really have this problem, but leaves will often be lost in the wintertime. So some plants will lose their leaves in the winter, which means fewer stomata, and thus a decrease in the rate of transpiration overall. Now, these are plant factors that will affect the rate of transpiration. If you have more leaves, you'll have more stomata, and thus an increased rate of transpiration. Your number of stomata will increase the rate of transpiration. If your stomata increase in size, the diameter of the stomata overall, that will increase your rate of transpiration. The position of the stomata, if they are positioned on the underside of the leaf only, that will decrease your rate of transpiration. If they are positioned on the underside and the top of the leaf, that will actually increase the rate of transpiration. Now, the reason for this is actually fairly simple. Water vapor rises, and if you're on the top of the leaf, it's fairly easy for water vapor to rise out of the leaf from the top of the leaf. So a cuticle is a waxy layer on the leaf that prevents the loss of water. A thicker cuticle will prevent more water from leaving the leaf. So a thicker cuticle will decrease the rate of transpiration. Now, this is a stomata. A stomata is just an opening in the leaf. A stomata is flanked by two guard cells. A guard cell will change shape, and the stomata will open and close dependent on turgor pressure. The fluid in the vacuole of a guard cell, when a guard cell is turgid, that will cause a stomata to open because of the radially oriented cellulose microfibrils. That will cause a guard cell to open, and a stomata will have an open position when the guard cell is turgid. So